Hello everyone, this is Anna, and we are now going to talk about part 2 of our histology section. And then now we're going to focus in on the stratified epithelium types. Alright, we're going to focus first on stratified squamous epithelium. And this particular image is showing you stratified squamous of non-keratinized epithelium. We're going to ignore this little arrow. When my PDF came over, a bunch of arrows got moved around and they're either disappeared or in the wrong places. So with epithelium, remember the first thing you always look for is the free edge. Okay, so this is a free edge, so we know it's epithelium, and this is the basal edge, and we know that down in here is where you're going to find your connective tissues. Now, if you remember back to part 1, I talked about how the name comes from the functional cells that are at the apical surface. So if you look at these cells, they are flat in shape. So these are squamous shape. If we look at the basal cells here, let me change the color, they are cuboidal in shape. And remember, these are the mitotic cells, but they are not the cells that define the function of this tissue type. And that's what we care about, is the function of the tissue type. So the function of this tissue is to resist abrasion, and that is what I want you to memorize with stratified squamous epithelium non-keratinized and stratified squamous epithelium keratinized, and I want you to memorize that. I want you to write it 10 times. So the way this works is the basal cells go through mitosis and they slowly get moved up to the surface. As they get further and further away, they begin to die, all right? This is less noticeable in the non-keratinized cells. It is definitely a feature of the keratinized, okay? Non-keratinized is going to be found in basically wet membranes. All right, so big idea, resist abrasion. Please do not ever write that stratified squamous epithelium protects things, because everything in the body protects things. I want to know more, okay? Let's look at a photomicrograph. All right, this is a photomicrograph from skin. So this is going to be a keratinized version, and I'll say this again in the future, but with keratinized, what you see are layers of staining. All right, whereas with non-keratinized, that doesn't happen because you don't have the same things happening structurally that causes differential absorption of the stain. Okay, so here is the stratified squamous epithelium. You will see the cuboidal cells here, and you will see the squamous cells here. And in fact, they are so flat that they are squished together, and all you can see is this flakiness, okay? And so that's been pushed up from here up to here. And again, function that you need to memorize, resist abrasion. So the previous slide was keratinized. This is non-keratinized. So with non-keratinized, because the cells aren't dying in the same way and building up the same structural features that helps it resist being in a dry environment, all of these cells absorb stain equally. So you have no layering. So you can see this is the stratified squamous epithelium. Down here, you've got more of the cuboidal shaped cells. And then here, you can see nicely how these cells have been flattened out. Okay, and they get flatter and flatter. So here you can see they're less flat, and up here they're more flat as they get to their functional surface. Okay, let's look at the next slide. So remember back when we were in part 1 and I talked about how there was a weirdo amongst the simple epitheliums, the pseudostratified? Well, transitional is the weirdo amongst the stratifieds, okay? I want you to never call this or confuse it with either of these. It is not stratified cuboidal, and it is not stratified squamous. There are some books that will describe transitional as a type of stratified squamous. We are not going to go there, okay? We're not going to go in and explain how this is true or not true, or talk about the exceptions and how weird things are, because you have enough to memorize. So what you're going to memorize is that this type of stratified tissue is called transitional and nothing but transitional, okay? So it does have multiple rows. The basal cells are cuboidal or columnar, but the surface cells, when they are not distended, not stretched, are a dome shape. Technically, when it's stretched, it will look squamous, which is why it sometimes gets confused with stratified squamous, but don't go there. Just don't worry about it. So if we look over here at the picture, actually let's look at the diagram first, or the drawing first. So here we have the mitotic cells, and they are roughly cuboidal-ish in shape or columnar in shape in some cases. This is where you're going to go through mitosis. These are not the functional cells of this tissue type. Remember, the free edge, the apical surface, is where the functional cells are located. When these cells are not distended, they are a bubbled dome shape. So cuboidal cells are geometric. Transitional cells are bubbles. They are not particularly geometric, and they can be kind of wonky in their shape. Okay? So we're looking at the diagram. So here on this picture, this is the transitional epithelium. This is where you've got connective tissue proper. These are the basal cells, and you can see the gigantic dome-shaped apical cells that are going to be doing the function of the cell. So with the name of this, I want you to memorize that 1, it allows distension without tearing, which basically means this bubble shape can be pulled into this shape. And then you would have another one over here. And it basically allows the bag to expand with less opportunity for tearing. So this is why it's dome-shaped. The second part of the function is about the layers. So it's multiple layers, it's stratified. It is stratified because the apical cells can be destroyed without you starting to bleed. You can sacrifice these cells because these mitotic cells are going to get pushed up. This allows it not to resist abrasion, but to resist chemical burning, because the primary place where you find this is in the urinary tract. So this is the bladder and the ureters. Those are the primary 2 places where you're going to find the transitional epithelium. Let's look at some more pictures of this. All right, so here, again, transitional epithelium, multiple layers, and, they are going to be a domed shape. Whereas, remember, cuboidal, if this was a cuboidal cell, it would be shaped like that. And then you would have layers like this. But it's not. These are bubbles. So they are dome-shaped or apical shape. Okay? And again, this means, well, when it's non-distended, it allows it to stretch without tearing. Okay? It allows the filling for the urinary bladder. So the reason I like this particular diagram is right here we have the drawing, but right over here we have the photomicrograph of this same drawing. So you can see this cell is the same as that cell right there. So it's a really nice way of kind of seeing the dome-shaped cells. And then down here it's a little bit harder to see, but you know, you can kind of use your imagination and you can see the basal-shaped cells. So again, location, ureter and bladder. Sorry about this arrow, ignore it. This happens when we do conversions to PDF sometimes. All right, let's go ahead and look at the next photomicrograph. So this is a picture that's likely to show up on assessments. So this is the lumen of the tube, lumen of the tube. If this was a living specimen, since this is the bladder, this would be urine touching the area. So again, free edge. So you know that this is epithelium. So this is connective tissue proper in here. We won't get into what type right now. You can see the sort of basal cuboidal-ish shapes up here. They're not real clear. And then you can see the doming right here of the apical cells. So multiple rows, and the apical cells are round domes or bubbles. All right, functionally, the multiple rows resist chemical burns, whereas the shape of the cells resists tearing when stretched or when distended. Okay? All right, next slide. Let's move on to looking at stratified cuboidal next. All right, this slide is the stratified cuboidal slide, and some of the words got cut off funky, so I just added it to the top. So again, stratified, 2 or more layers. Whoops, that's not what I meant to happen. 2 or more layers, okay? Now, in this case, you're using stratified epithelium not because there's risk of abrasion or chemical burning, but because you need lots of stuff made and secreted. So tons of making and secreting. So here's some examples of places where you would find it. Now, looking at our diagram, you will see that the basal cells are cuboidal because they are mitotic. But look, this is our free edge, and these are our apical cells or functional cells, and they are also cuboidal in shape, okay? If we come over here and we look at this salivary gland, it's a little bit hard to see because of the way the thing has been cut. We find a good spot. You can see roughly cuboidal-ish shape here, and then roughly some shapes in here. Okay, and it's not a great cut. Here's some flat cuboidals there, and then we've got some cuboidals right here. Okay, this is my ovary. Well, not mine, somebody's, probably a cat's actually. And so this is the oocyte, which will become the egg. So again, free basal edge right here. So those are the mitotic cells. This over here is the free edge in the lumen, and you will see that the apical cells are also cuboidal in shape, and they're all about making and secreting. All right, let's look at the next picture. All right, now we're going to look at stratified columnar, and the name also got cut off. Oh, columnar. Stratified columnar, okay? Now, what you're going to notice is the basal cells are also cuboidal, columnar in shape. They are mitotic. Okay. The surface cells, the apical cells, are the functional cells, and those are going to be columnar. Now, remember, columnar cells and cuboidal cells are functionally similar. So this just means even more secretion. So it makes and secretes even more than simple columnar. Okay. So if we look at the basal cells, you will see they are cuboidal in shape, and those are going to be the mitotic ones. The columnar shaped is the next row. Okay. Down here, we've also got cuboidal mitotic cells, and then we have columnar apical cells. And this would be your free edge, and this is the free edge. Right here is a lovely, what letter is that? Goblet cell. Okay. Stratified columnar is not commonly found in the human body. I actually have very few slides of it. Most of them I've had to borrow from the internet. We don't have good pictures of it in our AMP lab. This is a hint. It is hard for me to find good slides to show these to you and to ask questions on them, okay, because I don't have that many pictures. Look at the last slide, and the last slide is important because what I'm doing is over here, I am showing you stratified columnar, and over here I am showing you pseudostratified ciliated columnar epithelium. The key to differentiating these is, number one, how the nuclei line up. Okay? If I look over here, I will see a row of nuclei, and I will see a row of nuclei. They are neat and tidy little soldiers all standing next to each other. This isn't kindergarten. This is the third graders. They know how to stand in a line. All right? PCCE, those are the kindergartners. I have no line for the nuclei. They are all over the place. Down here, same thing. I've got nuclei all over the place. So no neat rows of nuclei. That is the number one way to tell the difference between the two of them. Okay? The other thing to keep in mind, and this was the hint from the previous page, I don't have very many good pictures of stratified columnar. So if I'm using a picture and you aren't sure because you don't see neat rows, when in doubt, you're looking at PCC or PCCE or PCE, because PCCE is very common in a lot of the tissues that we look at in this course. Stratified columnar is not very common for us to look at in this course. So you really want to know PCCE. You want to be able to recognize it. You want to tell me the functions of it. Stratified columnar, I don't care so much about, okay? PCCE, PCCE, PCE, those two are very, very important. All right, that is the end of part 2 on the histology videos, and I'm going to stop here. Part 3, we're going to do a concept map, comparing and contrasting simple epithelium and stratified epithelium, and working through the mental pictures you need to be able to distinguish these different tissue types. This is where I will be modeling how to think about it and how to start doing concept maps, because a lot of y'all probably haven't done very many in your previous courses, and it's an incredibly important skill to learn how to do in anatomy and physiology courses. All right, so we'll stop there.